Welcome to this week's Short Clip Friday podcast. Each Friday, I'll be posting 10 to 15 minute segments from previous podcasts. And the point here is just to bring you some of the highest yield topics from each podcast in order to kind of reinforce those topics. This clip is going to cover amino acid mutations and is from the very first podcast that was done on amino acids. Enjoy.
So on the MCAT, mutations will be presented in a certain notation. And if you have done any kind of lab research on amino acids or done lab research on different proteins, you're likely to have seen this. However, if not, you may not have seen this. It's going to be presented in the form of a letter, a number, and then another letter. So it's going to give the first letter corresponds to the amino acid that is normally present within the protein. The number is going to give you the position at which this mutation occurs. And then the third component, which is the second letter, is going to be the amino acid that is substituted in. So let me give you a quick example. So, for example, K285D, that would mean that lysine, K, at the 285th amino acid in the protein sequence has been mutated to a glutamate, which is D. It's pretty easy, but important to go over. And mutations can affect a lot of different parts of a protein. I'm going to go over a few different ways. This is definitely another very common question on the MCAT. So the first way that a mutation can affect a protein is when this mutation affects the substrate pocket for an enzyme. And so one of my podcasts is actually going to be just on enzymes. So all you need to know right now, if you don't know, is that a substrate pocket is where a molecule will bind to the enzyme, and then the enzyme itself catalyzes a reaction which spits out a product. So the substrate pocket is where that reactant binds. And so the substrate pocket itself can be involved in a number of different things. It can be involved in stabilizing the substrate and keeping it in the substrate pocket, or it can be even involved in the reaction itself to convert that reactant to the product. And all these functions of the substrate pocket are based on its amino acid makeup. And so a lot of times you'll have these different R groups in the substrate pocket which perform many different functions. And so I'm just going to list off a few of them for you. The first is they can act as acids or bases. Is a pretty important factor, especially for reactions. It can also act, these R groups that are in the substrate pocket can act as nucleophiles. They can stabilize the transition state of a reaction, can activate water by deprotonating water. It can activate some cofactor. It can act as a primer. It can form a radical. And so that's just a few examples, and these R groups are very important. And so when you have a mutation, that can really affect the functionality of an enzyme. And so I'm going to go over a little bit of how that happens. And so a mutation that basically changes an amino acid in the active site from polar charged to nonpolar is likely to affect the enzyme's function. So you can imagine an OH group in the substrate pocket, right? And so say that acts as a water deprotonator. And so if that were to be mutated, let's call it a tyrosine. Let's say that tyrosine was mutated then to an alanine, and now you're losing that function there, and your enzyme might not be functional at all. However, the exact reverse could be true too. So let's say you have an active site tyrosine, like I was just talking about, that OH functional group sticking in the inside of that substrate pocket, and you have a mutation in which you mutate that tyrosine to an alanine. But you then measure the activity of that protein and nothing changed. So what exactly does that mean? Well, that probably means that amino acid did not have much to do with catalyzing the reaction. So maybe that has to do more with the structure of the actual pocket itself, and substituting those two amino acids didn't have anything to do with changing the shape of the substrate pocket. But that's just evidence that OH group, originally from tyrosine, may not have had anything to do with catalyzing the actual reaction. It's important to note that cysteine is another amino acid that can act as an acid or base, and that it acts at a lot of the active sites of proteases, which cleave proteins into smaller fragments. It's important to note that non-polar amino acids do play a role in the substrate pocket as well, and so they can stabilize different molecules while they're in the substrate pocket. To give you an example, phenylalanine, which has that big phenyl ring, which is a benzene ring, can interact with other nonpolar amino acid residues via van der Waals interactions, also called hydrophobic interactions. Other big bulky groups with lots of carbon and hydrogen can interact with these phenylalanines and kind of stabilize this reactant in the substrate pocket. And outside the substrate pocket, mutations can also kind of mess with the protein, and we'll get into that next, of how these mutations can affect stuff outside of the substrate pocket of a particular protein. All right, so mutations can also affect the overall three-dimensional structure of the protein itself, not just that of the substrate pocket. So next, I'm going to talk about how amino acids play a role in determining the overall three-dimensional structure of proteins. You may have heard this called the tertiary structure of a protein. So just know when I'm referring to the overall structure, I'm talking about the tertiary structure. Before I get into the mutations that are going to affect protein tertiary structure, you should know that there's a few very important amino acids in determining this overall structure of a protein. So the first is cysteine residues, and a residue in molecular biology refers to a specific monomer within a polymer chain. So in this case, it is a single amino acid. And so these cysteine residues can form disulfide bridges that basically link covalently different parts of the peptide together. And then it's also important to note that hydrophobic amino acids will tend to be buried at the center of a protein, while hydrophilic amino acids will be located on the exterior of the protein in contact with the aqueous solution surrounding that protein. And this is simply because the hydrophilic amino acid residues prefer aqueous solution. And lastly, salt bridges can also form that support protein structure. And so salt bridges are just a link between electrically charged acidic and basic groups, especially on different parts of a large protein. And so mutations can affect these in a few different ways. Obviously, if you have a salt bridge in your normal enzyme, and then you mutate an acid that's away from the substrate pocket, but you mutate this amino acid from a basic group now to an acidic group, now what's going to happen? So now you have an acidic group and an acidic group that this mutated basic amino acid used to be attracted to, but now you have like charges, and those are going to repel, and that is really going to do a number on the stability of your protein. So as you heat it, this thing's gonna break right down. And I'm gonna give you a quick example of this concept, kind of and tie everything together. So you have pepsinogen, which is a peptidase in the stomach, helps break down peptides, makes them easier to digest. So let's say that this peptide undergoes a K25D mutation. This is the notation you're going to see on the MCAT. And so to decode that, you have K, that's lysine. So you're going to say lysine, the lysine in pepsinogen at the 25th amino acid position was mutated to a aspartate, which is D. And let's say that because of this mutation, you know that when this enzyme is heated, it becomes dysfunctional at a much lower temperature than the wild type,
or the non-mutated enzyme. And so it's going to say, okay, what does this mean exactly? And so if you think about it, the K or lysine has a positive charge at physiological pH, and that should make you think, okay, maybe salt bridge. And so now let's look, what did it get mutated to? It got mutated to D-aspartate, which has a negative charge at physiological pH. So this specific amino acid or specific residue within the protein went from a positive charge to a negative charge. And at this point, you can kind of take all the information they've given you, put it together. You know that you have a change in charge. You know that your enzyme became less stable. And you can start to conclude that the amino acid at position 25 was involved with a salt bridge. Furthermore, what's likely going on is two charges. You have a minus from aspartate, and you have a minus presumably from some other residue in the amino acid. And these like charges repel each other, and your protein now is less stable. Boom. And it's also important to note that mutations in cysteine residues can also affect protein folding and enzyme activity. These, I like to call what's wrong with my enzyme problems, come up quite a bit on the MCAT, and they're kind of fun, make you feel a little bit like Sherlock Holmes, and a little investigative. They'll give you that little aha feeling when you figure them out. But just keep in mind these come up quite a bit, and I'll talk a little bit more about these kind of problems on the Enzymes podcast. And so if your protein or your enzyme has some kind of disulfide bridges, then if that cysteine becomes mutated and you can no longer form that bridge again, you're going to have a protein that's less stable. And so a lot of times on the MCAT, how stability is measured is protein is heated up, or an enzyme is heated up, and its activity is looked at as a function of temperature. And so an enzyme that's become mutated will typically degrade at a lower temperature than, say, the wild-type enzyme that doesn't have certain mutations, or an enzyme that has mutations that do not affect its overall protein stability or folding.